Thank you. We're gonna wait a few minutes. We'll let people trickle in and get settled. For people in the room, we've tried to run some extension cords since I imagine most of you will be using laptops. If you use the plugs in this wall here, be careful not to create a tripping hazard, is the one thing I wanted to say. And there are some plugs in the back corner, but we aren't sure if they're working, to be honest, so. It has a what?
Getting jammed in it, like one of them's broken.
Oh, yeah, okay. Okay, so maybe don't try the back corner. Okay, I'm gonna start with some introductory stuff so that if people are still trickling in, they aren't really gonna miss anything super important. Welcome. Excited to have you all here and to teach you about pupillometry. I'm going to start with some acknowledgements. So first of all, funding for this project comes from my Marie Curie postdoctoral fellowship. So this is how we're paying for food for this event. So yay, Marie Curie, thank you. And also acknowledging BCBL, which is hosting this event. We are going to be getting a lot of lectures and talks from me for this workshop, but I have also invited some wonderful, talented pre-doctoral students who are going to lead some of the sessions. So Daphne Weiss will lead a session today where she'll tell us about Experiment Builder, which is wonderful because I know nothing about how to use Experiment Builder, but it is the iLink proprietary software. And so I wanted to include that as an option for people who would prefer to use that for building their experiments. So Daphne has prepared a wonderful intro to that. Marco will be leading the intro to R session this afternoon. I've marked that as optional on the calendar. The meaning behind that being optional is, I mean, anything for this workshop is optional. But in that case, I just wanted to note that that session it's good for anyone who's less experienced with R or maybe just hasn't used R in a while and needs a warm up to remember all of the syntax. So Marco's created a really wonderful, fun, interactive session for us this afternoon. And then Julieta Laurino was here with us up through September of last year. She's now back in Argentina. She'll lead a session tomorrow remotely. That session will be a deep dive of how she used growth curve analysis to analyze her pupillometry data. So she came last fall and I pointed her towards things to read to learn how to do growth curve analysis. She taught herself and she did an amazing job on her project. So I've asked her to show that data set as like a real example of applying that kind of analysis to real data. So that should be great. So some wonderful invited speakers here. And also there are some wonderful volunteers who have agreed to help me set up food and various things during the event. So a big thank you to them as well. I have sent emails out. If you aren't getting email updates, then you're probably not on my email list. So you can let me know or talk to your neighbor and maybe they can forward you the emails. If you have not already, there is a Google Drive that has all of the files for this workshop. So that's things like slides that are going to be presented to you, lots of code, because this is a very practical workshop. My goal in this workshop is to give you everything you need to start your own pupillometry projects and to make that as easy as possible for you to start your own pupillometry projects. So I'm giving you all of the code I actually use to analyze my real data. I'm giving you examples of my real data that I have collected. I'm giving you example experiments that we will go through, and I'll tell you how you can take these example experiments and turn them into what you need, because we're all doing slightly different things. A lot of the examples in the workshop will be speech perception-y because that's my research area. But I will try to also generalize this a bit because pupillometry, as we'll see and talk about in this first session, is a tool that you can use for all sorts of different cognitive and neuroscientific questions. So it's not just for linguistics. I just happen to do a lot of linguistics. Post-workshop, you'll be able to find recordings of these sessions on my website. So if you miss a session or you maybe feel like when you actually have your pupil data, you're going to want to go back and rewatch a session because you'll understand better what you need to get out of the session. They will be available. I'm also going to make them available for BCBLians on the wiki. Forgot the word. And I'm going to essentially maintain the Google Drive in its current state, or essentially we might add a few more files throughout the workshop. For the most part, it will remain unedited. So people who come back and watch these videos will have the exact same files. But I'm always finding new ways to make my code more efficient or better or add new things based on current research. So I will always upload my most recent versions of the code to my GitHub. So if a year from now, you're coming back to this workshop, you're watching one of these videos, so I'm speaking to the future right now, future people who are watching this. If you want to, you can look at the GitHub and you can see if any updates have been posted. And that'll just give you an idea of like, maybe Drew discovered that there's some additional processing step that should be done, and she's added that to the pipeline. Okay, that's sort of my broad introduction. Do we have any questions before I get into the actual lecture? Okay. Let me switch slides real quick. Okay. So, day one, lecture one. This is our, what is pupillometry? What do you need to know about it? How is it going to be useful day? So starting out with this first lecture is going to be key information about the evoked pupil response. We're focusing very much so on cognitive pupillometry, which I'll go into in a bit more detail in just a moment. But I also want to take some time to talk about anatomy today, because these are basics that if you're going to use this method, you should be aware of. At the very least, it'll be a list of citations that you can put into your own method section in your future paper. There will be a coffee break today. There will be food and drinks in the lounge. And then we will do another short lecture. Well, I'm going to go into a little more detail on current best practices. Things we do and don't know about the pupil will also be quite a bit of a theme in this lecture. For this workshop, I'm very much a pragmatist. So we're doing lots of what I call practical examples and actual R tutorials. And you're highly encouraged to download the materials and look at them on your own laptops while we're going through this. The first practical example will be led by Daphne Weiss, and it's going to be an example of how you could use Experiment Builder. And there won't be anything you can actually open on your computer for that one, unless you want to look at the slides. But the second practical example, which I will lead, will be going through a CyclePy sample experiment. That's something that if you have CyclePy on your computer or really any code viewer, you can open that sample experiment and look at it while we are going through it. You don't have to, but just wanted to let you know that that option is available. There is not a lunch provided today. So for those of you who are interested in that optional intro to R session, you should come back at 2 o'clock in the afternoon, and Marco will be leading that. And if you are not partaking in that, then your day will end nice and early. Tomorrow will be the lots of R code day, and we'll take it one step at a time. So even if you're newer to R, hopefully it will not be overwhelming. And so you're highly encouraged to bring a laptop tomorrow in particular. So let's get into pupillometry basics. This is a picture of equipment. We aren't going to talk much about the hardware. I highly encourage you to, if you're here at BCBL, we have iLink eye trackers. There is lots of resources provided by SR Research, the creator of those eye trackers that we have access to. That's all about the equipment. The equipment is also already set up in all of the rooms. And if you talk to Effie or I, we'll be able to tell you the details about equipment setup. So we aren't going to cover that today, but I did just want to show a picture of what this looks like. So this is our actual eye tracker setup at Corta. Yeah, this is at Corta. So this is the eye tracker camera, and it is currently pointed at Daphne Weiss, who is my victim of this picture. She is looking at a computer monitor that looks pretty boring. It just has a fixation cross in the middle. And that's how a lot of my pupillometry experiments are actually set up. But we'll talk a bit about how you can do things with images and with text. But some of the considerations you need to take into account. Because I do a lot of speech perception, I'm sort of lucky in that I can just put a fixation cross on the screen and not worry about all those other details. But we'll talk about what to do if you do need to worry about those sorts of details. So what the eye tracker does is I don't understand the technology actually on a deep level, but it is really amazing. It is able to, with all of this additional information around the pupil, it is able to laser in on that pupil by gathering information about infrared light and identifying where the iris, the sclera is the white of your eye. And it's able to essentially hone in and get a read on where your pupil is. The eye tracker, given its name, is also used for tracking gaze position on the screen. And it is incredible. It gives you pixel information about where someone is looking at on the screen. But if you have an eye tracker and you know how to do eye tracking, you know how to do pupillometry to an extent, because it is the same exact equipment. While this eye tracker is keeping an eye on the shape of the pupil in order to determine where you're looking on the screen, it is also tracking very small changes in the size of the pupil. So the eye tracker is set up to automatically always record X and Y position and the size of the pupil. So if you've done eye tracking experiments, you will have a column in your data that is the size of the pupil, almost guaranteed. I don't even know if you can turn it off. That doesn't mean that pupil data is good quality. If you didn't design your experiment with that in mind, it might not be good quality, but the data is actually there. So there's an animation I'm going to play. It's very small because these changes in pupil diameter are very subtle, but this is what, with this technology, we are able to keep track of. So let's get some definitions out of the way. Cognitive pupillometry. This is the measure of the pupil diameter over time. We often call it cognitive or task-evoked pupillometry when we use it in experimental research, because we are quite literally evoking a change in the size of the pupil. So this is an evoked response in that way. It's really similar to the language we use when we talk about EEG as well. It's used in a huge variety of cognitive research areas. I use it in linguistics, which is why I have this comment that it's not just linguistics, but it's used in memory, attention. It's used in sort of like criminal psychology work as well, because the pupil changes in size for a lot of reasons, which is one of the things we're going to really talk about in this session to clarify how we can measure the cognitive pupil response when actually the pupil responds to a lot of things. Why we're tracking the pupil. There is a systematic relationship that has been demonstrated between increases in pupil diameter and cognitive load, which I am going to be defining as the amount of cognitive resources that are being recruited and allocated for a cognitive task at a given moment. And I'll explain why I like this definition personally. There are lots of papers out there, especially in my research area in speech perception, that call the size of the pupil an index of effort. And I actually don't love that language. And I'm going to explain in this lecture why I prefer to think about it as recruitment and allocation of resources. I want to start with a guide to how to interpret a pupillometry plot, just because you're going to see a lot of these. So first of all, you're going to see lots of plots with these nice little curves on them. On the y-axis will always be your pupil diameter. It's often just labeled as pupil diameter, but it is actually a measure of change in pupil size. So it starts from 0, and we'll talk about how we baseline the pupil, which means we get the size of the pupil before a trial starts, and we usually subtract it from the rest of the trial so that what we plot on a plot is actually a change measure. And so usually this is the change in your pupil diameter here on the y-axis. And then we typically, when we're presenting data from these types of experiments, we line it up relative to trial onset, or more specifically, typically a stimulus onset. So in a lot of my research, this is, if they're listening to sentences, this is the start of the sentence is 0 on my x-axis. And that'll be clarified almost always in the captions. We are not just measuring the pupil for one type of condition. Typically, the reason we are tracking the pupil is to compare different conditions that we have created in our experiments. And by doing this, we are then able to draw conclusions about the cognitive challenge imposed by different conditions. So, for example, if I had collected data for my experiment, and if I had designed it following a lot of rules that I'm going to lay out for us, then I would be able to draw the conclusion that the light blue line at the top is inducing more cognitive load on the listener than the lower dark blue line. So that is the end goal in the design of our pupillometry experiments, is to be able to leverage the pupil response to draw those types of conclusions. One last thing, just to reiterate what's going on in this plot. In my experiments, if 0 is the onset of a sentence, and sentences typically end a little bit before the peak of the pupil response, the highest point of this amplitude. What we are looking at, so this red line is going to go across this plot. So if as we go across this is 3 seconds of time, 3,000 milliseconds. So we are looking at this change in pupil diameter over time. And now if you look at the left where the pupil is, this is a bit of an exaggerated plot, but this is to show what we just saw happen was an increase in pupil diameter, followed by a little bit of a decrease. This is really common in the evoked pupil response. When the task itself, when the stimulus is happening, we get this evoked response, and it usually, after the stimulus, after the cognitive demands of the task die off, it starts to taper back down. If you have a task where the demands do not stop and it keeps going and going and going, that doesn't mean the pupil is just going to skyrocket forever. That's not possible. And that's actually why in all of my experiments, I tend to sort of say, like, the maximum I've ever done is 2 sentences in a row. And you'll see some of the data from that in this workshop. It was collected by a master's student named Jimena, and she did an excellent job. Some people are interested in narrative length materials, and I have done peer reviews of some papers where people are using pupillometry over really long time periods. The data is a little bit different when you process it that way. And so we're really going to be focusing on this type of an evoked response for this workshop. Okay, so, and just to reiterate, if my experiment is listening to sentences, the conclusion I would be drawing is this dark blue line, whatever condition that was, those were the less challenging types of sentences, and the light blue would be the more challenging types of sentences. Yeah. Yeah. So David is asking, I'm just going to repeat questions. David is asking how quickly the pupil responds to the cognitive load. There is understood to be a 200 to 300 millisecond delay in the pupil response. I have to say in my own data, it typically looks like 300. But what I have seen written in papers is 200 to 300, and yeah, we'll discuss a little bit about how there's a bit of a lack of pupil methods research. So I don't have a good citation for that. It's just the number that everyone reports in their papers. Yeah. And also feel free to ask questions at any point. This isn't meant to be hold your questions to the end. This is welcome to be interactive. So I encourage you to ask questions as we go. Okay, let's talk a bit more about some key information about this evoked response. I'm going to start with a citation that is in just about every cognitive pupillometry paper. So this is the first really big empirical work that used pupillometry. It's work by Beatty that was published in 1982. This paper is like a tour de force. They do everything in this paper. It is like a dozen experiments written up in this short little report. They don't even really go into details of the methods, which drives me crazy, but it is so much in this paper. And essentially the goal of this paper was to demonstrate that this pupil response is a reliable index across many different areas. So they did memory experiments, they did attention experiments, they did lots of language experiments, which was great for me because I can go back to this paper anytime I want to see how language compares to other cognitive research areas. So this is some of the data from their short-term memory task. So this was a digit span task. So what you are seeing here is the evoked pupil response when participants had to remember 3 digits, 4 up to 6 digits. And so we can see there's a very systematic relationship where in the order of the digits increasing, we are getting a larger amplitude. You might be looking at this plot and thinking, wow, that data is not very pretty. That plot is sort of, it's not just pixelated, it's like really like jagged. Well, this is from 1982. So, I mean, partly there's a little bit of pixelation from the original print quality, but also this is back in the day before they had things like the iLink. They were taking pictures of people's pupils, printing them out and measuring the size of the pupil at all these different time points. And so it is especially impressive. This paper just amazes me in hindsight. They have like a dozen experiments that compare across all of these different domains, and they were doing it without the technology that we have now. This is also the reason there's a bit of a gap in the literature, like from this enormous important paper to like basically early 2000s. There's not a lot because even though they demonstrated that this tool is really cool and it could really be useful in the kind of research that we want to do, you can't really use it if the amount of man hours it takes is that cumbersome. And so the dawn of the iLinks and Toby's as well is really what has changed and propelled pupillometry research. The last thing I'll note from this paper is they have this lovely plot where they essentially put out all of the different conditions and tasks that they did divided by categories. You can see they have memory, language, reasoning, and perception here. This was the peak, so the largest pupil size recorded on average in trials for each of these tasks and conditions. So we can see first, if we focus on the memory side, 1, 2, 3, up to 7 digits, we can see that very systematic thing of stacking, that hierarchical relationship, just like in the plot I was showing you before. This is what we call a summary statistic, essentially. We were looking at this curve of data. They have taken just the peak value here, and that's what they're representing for the 7-digit value. And we'll talk more about that tomorrow as well, peak measures. You can see also slipped in here, there's a 4 words. So I guess they had another memory task where people had to remember 4 words. And so what's really nice is you can see how these different tasks stack up. What I will say is I think we're sorely overdue for a repeat of this sort of tour de force doing everything all in the same data set. Because as I was saying, this was before we had the technology that we have now. And so you can imagine if we went back and did this, we would be able to make this same chart comparing across all of these things, and the data might be, you know, a little more reliable. But it's still a really nice reference. So I highly recommend this paper. And this is a good point for me to note that in the Google Drive, there is a folder called recommended readings. Anything that gets cited, put up on the screen in this workshop is in that folder. It's like 20 papers. That's not a small amount of papers, but hopefully you'll know what to look for in each paper, and that should save you some time when you're pulling together your own pupillometry paper. All right, let's talk a little bit about why the pupil correlates with cognitive load, because this is sort of funky. BD came out and was like, guys, we can use the pupil to draw conclusions about how hard tasks are. Look how reliable, how this index is really consistent. And even now, like I said, skip 20 years, and then in the 2000s to now, there's been tons of pupillometry research also showing it's a very reliable index, which is super cool, but it plagued me for years, just this bothersome in the back of my brain when I was using pupillometry, like, why though? Like I had heard like discussions of like, when I would bring this up when I was giving a poster at a conference, people would ask me, is there something adaptive about that? Is our pupil getting bigger because we're doing something hard and it's like somehow going to help us that our pupil is bigger. And I'm going to go through the anatomy and explain to you that that's not what's happening. This is sort of a side effect of our neural anatomy connections. So there are some free stickers. If you didn't see them over near the doorway, which you are welcome to take one. This is a side view of the eye. So we're going to walk through some of the anatomy here. The iris in that, the logo that I've been using for the workshop is that pretty blue-green or brown or gray or so many different colors of eyes that we have. This is where the magic is sort of happening because the pupil, the black of our eye, most people know what our pupil is, but it's actually sort of the absence of our iris. It's the center of the iris. And you can see that when we talk about the size of the pupil changing, it's actually going to be the musculature surrounding this empty space that is changing. The light that enters your eye goes through a lens. There's also more musculature that we are not going to go into about how the lens stretches and does things to like help you focus on different distances. It's fascinating, but we don't need to worry about it today. I had shown you earlier the white of the eye is called the sclera. It actually wraps all the way around. And then there is, it's being covered up right now by the subtitles, what's called the anterior chamber. So we have fluid. There's lots of fluid in the eye. And the other most important thing about the eye, and this is something that people who are doing vision research will of course be very interested in, is our retina, which is collecting light that enters the eye, interpreting it, and sending it along our optic nerve to our brain to be processed. The eye is a very miraculous piece of anatomy in our body. But let's talk about why the pupil changes in size. The paper that I'm going to recommend, which is in the recommended readings, is Strouch et al. 2022. I like this as an anatomy paper because it's pretty short. It's also fairly recent, so it's pretty up to date. When I was first starting doing pupillometry, because I've been using it for like 7 years now, I learned it at the very beginning of my PhD. When I first started using pupillometry, when I would look up why the pupil is changing in size, what I would see a lot of in papers at the time was, it seems to be related to activity in the locus coeruleus of the brain, or maybe it's a readout of like entire brain-wide activity, which are like wildly two different proposals. There has been more data that has come out in the last decade, which is great. And so this is a very nice summary of the current state of knowledge. I would say, if anything, the sort of the maps, like I'll show you the images in a moment, that Strouch and colleagues lay out are very conservative in their estimation of what is causing changes in pupil diameter. They call their paper Pupillometry as an integrated readout of distinct attentional networks, and I like that as a summary, because what they've done is they've created a taxonomy of where they sort all of these different responses into what they call low, intermediate, and higher level factors. So for this workshop, what we're leading up to here is we care about executive functioning, using the pupil in our research to draw conclusions about executive functioning. That's what we're really interested in. But we cannot ignore the fact that the pupil actually, it changes size for a whole lot of reasons. And you'll see in a lot of papers when they talk about using pupillometry, they'll just say the pupil changes size in relation to cognitive load, which is something I've also done in my own papers, because for the purposes of my paper, while that may be true, while I may have controlled for all these other factors, that is not actually true. The pupil changes size for a whole host of reasons. This is an arousal response. This is how we gate the light that goes back to our retina in order to protect our retina from getting too much exposure to light. So these factors get divided up into low level, which is primarily the light reflex and focal distance. We'll talk a bit more about the light reflex in detail. Intermediate, alerting and orienting response. I like to point these ones out because if you're designing an experiment, and for example, when I present sentences over headphones to participants, if every trial, I designed my trial where I played a beep at the beginning, and it happened 2 seconds before the sentence started. we will get a pupil response to that beep. Does that mean the beep is cognitively demanding? No, not necessarily. That is an alerting or orienting response. And so this is why keeping these other factors in mind is going to help you when you're designing your experiments to avoid eliciting pupil responses to the things you don't care about, because we don't want lots of bumps in our data. We want the one clean evoked response that is just for our stimulus. So I'm not gonna spend too long on this. I really do recommend the Strauss paper. There's another paper by Mathot. It's a French name, so it's got like a little hat on the O. That's a 2018 paper. It's also in the recommended readings, and you'll see I have some plots from their paper later in this PowerPoint. Those are my go-tos. There was actually, I saw on Twitter this morning, so I'll share this somewhere. There was just a book, a pupillometry book, like literally just published. Like it popped up in my Twitter this morning and I was like, this is amazing. And Mathot is one of the authors on it. And I really think he's great. He's an expert in the field. So this taxonomy though, as I was saying earlier, this paper by Strauch and colleagues, I think if anything, it is under-specifying this circuitry, but I think that's good in a way, because as I was saying, when I was first starting in pupillometry, people were saying the pupil changes size because of brain-wide activity, and that's a little bit presumptuous, perhaps. We know much more about these low-level factors than the high-level factors. The low-level factors are very much changing due to sympathetic and parasympathetic activity. The locus coeruleus and the superior colliculus are also very important circuits. I'll show you a plot in just a moment. The higher level factors, the current state of data indicates that activity in the frontal eye field and anterior cingulate cortex via a superior colliculus mediated pathway are what are eliciting this cognitive pupil response. So let's back up for a moment. I want to explain in a little bit more detail the light reflex, because this is the one we know the most about. It is very well researched because of vision research. It has nothing to do with our cognitive research in a way. We have here, I've been showing you this plot of this, sort of pretty iris with the pupil in the middle. I want to imagine that instead we are looking at musculature. This isn't necessarily exactly what it looks like, but this is like the kind of way it typically gets animated in anatomy textbooks. There are two types of muscles that affect the size of the pupil, and they're actually in like sort of competition with each other. So decreases in light, so getting into a darker space, will elicit sympathetic activity, which elicits dilation via the dilator pupillae muscle. When you're in a dark room, most people know this, your pupil gets big. It's the gate to your retina. It's trying to let in as much light as possible because it's hard to see in a dark space. When you are in a bright room, when there's an increase in light, this will cause parasympathetic activity, which causes constriction via a different set of muscles. This is a lot like a camera lens, actually. It's really fascinating. So it's sort of a twisting set of muscles. And so this causes constriction via what's called the sphincter pupillae muscle. These sympathetic and parasympathetic pathways, they're antagonistic. And there's a lot of knowledge about this light reflex and how, like if you read into detail in the Strauch paper, it'll talk about how anytime there's a dilation of the pupil, there's a little bit of a correction. Where it also constricts a little bit because these two muscles are in competition and they're trying to essentially balance out. So that's just a little bit of the deep dive on the anatomy of what's going on. And I also just wanted to show you that side view again to really drive home that when we're talking about constriction and dilation, we're talking about that iris essentially opening up and letting more light into the eye. Now, the greatest part of the Strauch paper is this diagram, and I know you're looking at this and thinking that's sort of a mess. But actually, if you go into this paper, which I highly recommend, they have all of these different neural circuitries that they are essentially taking the current state of knowledge and distilling it down to what we have good confidence in. The most important things for cognitive research is activity in the ACC and FEF. These are in square boxes, and that's to indicate that they are that higher level executive functioning. You can see, the one thing I really want to point out in this figure, and you can go look at it yourself, the ACC and FEF do not directly link to the pupil. And this is the really important thing. This is why they are affecting the size of the pupil through a SC mediated pathway. So we can see the ACC here, there's a line pointing to the SC in red. The FEF also points to the SC in red. The SC feeds in to these different areas, and eventually it gets to the pupil. So the big takeaway for those of you who are itching to know why this is happening is this cognitive pupil response is sort of an incidental effect caused by activity in brain areas that are responsible for executive functioning. And long story short, we can take advantage of this effect because it is very reliable. But as I've been trying to really drive home in this lecture, you have to control for a lot of factors if you're going to correctly leverage pupil information. So you need to check your assumptions because the pupil changes size for a lot of reasons. Yeah, so this is just going back to those original taxonomy, which I do think is a very nice breakdown. How can we draw conclusions about cognitive load or effort, as it gets referred to a lot in my research area, when the pupil responds to just about everything? And the answer is going to be with clever and careful experimental design. Yes?
The increase in like the level of practice, for example, low level, do we get faster pupil response than high level practice, or it doesn't matter?
I do not know, actually. That is a great question. I'm going to jot that down. I'm going to see if I can figure it out. I'm going to keep track of questions I don't know the answer to and try to report back on the next days of the workshop. Okay. Yes, Manu.
In the slide before, you said that the pupil respond to changes in cognitive areas, higher level cognitive areas, or the other way around.
Activity, the current state of knowledge is the ACC and FEF. Activity in these areas will, via the SC, prompt changes in the pupil diameter. Does that make sense? That's the direction. So the arrows here are informative in this figure. Okay. Now we're getting to a part of this lecture, which is one of my pet peeves, actually, about pupillometry. And in my research area in particular, a lot of the time, the pupil response is reported as a measure of effort. And I'm going to explain to you why I don't like this, because to some degree, we're just choosing terms to describe this very complex thing. And it's reasonable to choose the word effort in a lot of contexts. But I want to show you one paper as an example. This is work by Chu and Braver. In this task, we aren't going to get into too much detail about what their exact task design is, but the most important thing for this message that I'm about to convey is they were comparing pupil response for incentivized, these are paid trials, versus non-incentivized trials. So at the beginning of a trial, participants would be shown what's called an incentivized pre-Q, where they either saw a green box, which means they aren't getting paid for the trial, or they were shown a money sign, which means they're going to make money. They then did this, what's called an AX continuous performance task. Don't worry too much about the task. Just know that they are doing something that's going to elicit a pupil response. And we're going to compare the trials where they're given money versus the trials where they aren't given money. When we do this, what we see, and this is not just this Chu and Braver paper, this is lots of papers like this, we see there is a larger pupil response on incentivized trials. So that's these two red lines here are incentivized trials versus the non-incentivized trials. So if we were to say the pupil responses effort, that would mean that even though they're doing the exact same task, when they're getting paid money, the task has become more effortful. And this is why I don't really like this term. And instead, I like to characterize the pupil response as an index of resource allocation. And that is why it is impacted by a participant's motivation. So motivation has been shown to impact the size of the pupil response. If participants are more motivated, they are going to be more engaged in the task. They're allocating more resources. Does that make sense? So that's one of my biggest things, my biggest take-home messages is not to underestimate the impact of a participant's motivation in research and how we characterize the pupil response as well. Now I'm going to switch gears a little bit and I'm going to start talking about what I'm going to call nuisances for cognitive pupillometry. This is essentially all of those lower level factors that we were talking about from that taxonomy. If we care about the higher level factors, we don't want to elicit pupil responses for things like the pupil light reflex. If we aren't, anatomical researchers interested in how the musculature of the pupil works, which I think in this room no one is, then we don't actually want to study the pupil light reflex. We want to study cognitive functioning. So I'm going to show you some plots from the Mathot paper. This is the other, I think, really nice. It's a bit longer, which is why I sort of would say after you look at the Strauss paper, come back to the Mathot paper. But he goes through quite a bit of anatomy too. Let me move this real quick. Yeah. So this is a plot from Mathot's paper. He has some really nice, just basic plots to show things like what happens to the size of the pupil when you shine a red or a blue light at it. So there are two lines here. There's a red and a blue line that does correspond to the color of the light being shown. And we don't need to worry too much about it, but technically different colors of light. You can see that the initial pupil response is the same, and then they sort of taper off differently. Again, we're not going to get too much into that really, but the big thing here, this is an absolutely enormous decrease in pupil diameter. So again, just as a reminder to sort of pause, you shine a light at the eye, the light's going into the eye, and your pupil's job, partially, is to protect your retina. It constricts the gate of that light going to your retina, can get smaller because we don't want too much light to be hitting the retina. So we see enormous changes in pupil size when we shine light at the eye. What does that mean in a practical setting when we're actually running an experiment? Images of different luminance and different colors can be problematic. And now I'm going to make it clear in this lecture, it doesn't mean you can't do this. And in fact, there's a predoctoral student, I couldn't remember the word, a predoctoral student named Hadil who will be presenting some of her data on Friday. And she has converted the classic bilingualism switching task where they're being shown pictures and naming things. She has converted that for pupillometry and it is working beautifully. And so that clearly has lots of images involved. So this is just to say, this is one of those things that can be problematic and you have to very carefully design your experiments if you're using images or text. Yes, Marco.
Both the infrared light and the affects the responsibility to itself, and if that's like changing between one, they are with different infrared lights.
I actually do not know. It's another one. Marco was, for those online, Marco was asking if infrared light will elicit changes in the pupil. I will look it up and report back tomorrow. I do know that infrared is part of what feeds into the eye tracker system. They track infrared light, and that's partially how they identify the pupil. And so different shades of colors of eyes can sometimes be harder to track with the pupil. I haven't in practice actually noticed any issues with brown-eyed people, for example, but okay.
I have a question.
Yeah.
You're talking here about light, but does shape also matter? So suppose I'm only using black and white, but if I'm like only looking at a a circle versus a very complex shape of.
Okay, Sandy is asking about whether the shape of the image on screen might matter. Yes, if you have an image on screen that's, you know, maybe like 10 inches by 10 inches, there are a couple of reasons this can matter. First of all, if there is more luminance in one part of the image, if your participant is able to look to different areas of the image, then that luminance is going to be affecting the size of the pupil, yes. If you have a small image where they can't really do essentially search and things like that, then it will most likely not cause issues. Essentially, if you restrict the amount of room to look around, the luminance should be fairly controlled for. And so that's why if you do have images on screen, I do recommend them being quite small in the center of the screen. Yes, that's the other thing I recommend is matching the luminance across all of your images. But that being said, it can be really hard if you have a complex image, maybe the picture of a cat, and you have it really big on your screen, and maybe the background is white and your cat is black. If they're looking up at the head, is there more white surrounding the image up there than if they're looking down at the paw, or vice versa? So this is where, again, shrinking up your images and matching luminance are the best ways to control for that.
Follow-up.
Yes.
Would it then be the best way to control that, not show images whatsoever?
Sandy is asking if we should just not show images. That's been my strategy, actually. It's just, but I, like I said before, I am lucky in that I do speech perception, and in a lot of my research, I can focus on just purely auditory stimulus. And then I never have to worry about images. That being said, as I was just saying, as an example, Hadil will present her her work that she's going to present for her midterm also in a couple of months. And she's presenting images. It's the entire task is images, and not only images speaking, they are producing words. And I have never wanted to look at pupil response during talking. It just sounded too complicated, and it's been working really well. So it is very possible, for sure. Teza?
Yeah, what about the colors of the areas?
Yes. So the official iLink information is that some eye colors will be harder to track than others. In practice, I have not noticed this. It's not that anytime someone who's with brown eyes shows up, I suddenly can't track their pupil. I think bigger impacts on the quality of tracking of the pupil would be things like eye makeup, because if your participant's wearing a lot of mascara, the eye track will have a hard time, and droopy eyelids. Which is particularly in older populations. When I was collecting data from older adults, I ran into issues with that. You can sort of set up the eye tracker to sort of like have people like essentially like if there are ways you can sort of get the eyelid to not be essentially obscuring the pupil quite as much, but there's not always a solution to it. And sometimes you have to turn away participants who.
It is more about the anatomical, rather than tracking that the physiological response is different because some people is more sensitive to light than others.
That's a great question. Another one that I will look up. Yeah. Cesar is asking if different eye colors might be more sensitive to light. Yeah, I do not know the answer to that, but we'll look that one up. Manu?
Is there a relationship between eye movements, including microseconds and pupil dilation?
Oh, Manu, you're jumping the gun just a little bit, but let's skip to it. We'll back up to the number 2 here. Saccadic eye movement does, for 2 reasons, affect the measure of pupil diameter. So first of all, the image I'm showing here is looking at the 4 different corners of a screen, and this changes the shape of your pupil. It's very small. It's hard to even see it here, but it does change the shape of the pupil. Now, what I haven't seen done, but I would love to see done, because the eye tracker, the iLink at least, it can measure either diameter or pupil area. And I would love to see a methods paper compare those 2 for a visual world paradigm to see if one of those, if essentially switching to pupil area reduces this effect. The other thing, which I think is what Manu was getting at with her question, is that actually the saccade itself does elicit a small pupil response. I don't know about the distinction between saccades and microsaccades, to be completely honest, but saccadic eye movement elicits a pupil response. So Let's say you want to do visual world paradigm. You want to do concurrent eye tracking and pupillometry. Would I recommend this? Maybe not, but because also the images on screen, controlling for luminance and the background color, and when you're looking around, there's so many things that I have already told you are really problematic that come up with a visual world paradigm. But what I will say is that we'll talk a little bit about complex analysis approaches, and one of them is generalized additive mixed modeling. There is a paper that I will show you tomorrow that used, these are GAMs is what they get abbreviated as, showing how GAMs can be used for a visual world paradigm, and you can actually put the X and Y coordinates into your model to control for them. The other thing that I have not seen done, but I imagine you could do, because iLink does give you information about timings of saccades, you could probably feed that information into a model. So there are complex modeling procedures that could, in theory, control for all of the chaos in your data that's being caused by a visual world paradigm. But for example, for my postdoctoral fellowship, I had proposed a project where I was interested in eye tracking data, essentially timing of lexical access, and I wanted to do pupillometry. And rather than propose a visual world paradigm where I did both, I split it into 2 experiments. I have one experiment where I did essentially it's the same group of participants, but I have them do the task where it's a visual world paradigm, and then they do a task where it's just a fixation cross. So essentially I decided I just wanted to avoid the issue.
Yeah, but the problem is that the eye is never steady. So even with the fixation cross, the eye is always moving, otherwise you don't see it.
Yeah.
So that's the link between microsaccades. It's a big jump.
Yeah. Manu is pointing out that even when we're looking at a fixation cross in the middle of the screen, there are microsaccades because we aren't able to keep our fixation steady. No, it's an interesting point. That might be essentially a source of noise in the data that is minor enough that you can extrapolate away from it. It would be my response. We'll see a lot of pupil data tomorrow and talk a bit about estimating power is really hard. I haven't had any issues in my studies with the research questions I've asked so far, but we can talk more about that tomorrow when we're actually looking at data. Let's back up real quick because I did skip the nuisance number 2. This is the focal distance response. So this is, if you remember from the taxonomy, these were both low-level responses. And so Matho also has a figure of this where the participant has shifted their gaze to a nearby object and he's tracking the size of the pupil. And so what we're seeing here is a large decrease in pupil diameter. So changing the distance that you are fixating on also changes the size of the pupil. This one doesn't come up quite as often because when we're doing eye tracking and pupillometry, typically participants are looking at a screen that's a fixed distance. The one thing I have thought about in my own work is a lot of the times I will play a sentence and then at the end of the trial, the participant's job is to type what they heard. And so I am not actually interested in the pupil response during typing, but looking down to type and then going to the next trial, they are looking from a keyboard up to the screen. And there is going to be a pupil response. And so this is why we'll talk a bit about in design, you want as much time as you can squeeze into your experiment every trial, where the participant is essentially looking at something very basic, a fixation cross, or maybe a scrambled image, before you put the stimulus itself, because you want to spread out any sort of changes due to typing or whatever from that previous trial as far away from the onset of your stimulus. So this is the only case I've really seen it come up in my own work. Perhaps someone else will have thoughts of how it could come up in their work. These are the 3 primary nuisances. There are even more. I want to talk a little bit about how big of a nuisance these are, particularly the first 2. So these are the same figures I was showing you before, the pupil light response, the pupil near response. And what I put up here is in contrast, this is a pupil response elicited to hearing a beep. This is not necessarily even a cognitive response. This is probably an orienting response, which is often smaller than a cognitive pupil response, though it depends. This is to sort of show you the magnitude of these changes and also that they go in different directions. So we are going to look at some data tomorrow. This is a preview of that data. So just sort of keep this in the back of your mind. What happens when we have a huge change in luminance at the same time that we are eliciting via a stimulus, our cognitive response, is we get sort of an additive effect. And what I mean by this is some real data that was collected here at BCBL by my master's student. This data is fine. It is fine. But if I were to do this experiment again, I would change something slightly, which is I can see that before the stimulus onset, which is at 0, the pupil was decreasing in size. And I know because of how I designed this task that it's because there were things on the screen. There was a decision screen and they had to click on a button. And then it goes into the trial. I didn't leave as much time as I should have between that decision screen and the onset of my stimulus. And so what we see here is this is listening to speech. This is a cognitive pupil response here. This pupil response, we can still observe it, we can still measure it, and it doesn't ruin the data, but the data would be better. And in a future experiment, I would spread things out in time. So that is the #1 strategy I use in my work, is spacing things out in time as much as possible so that during this prior to stimulus onset period, the pupil will hopefully be a lot more level. Essentially, it won't be changing in size. So we don't have these competing effects that go in opposite directions. Daphne?
So an artifact like that, we found out afterwards that there was something else going on. Is that something you could detect with a very small pilot sample, or do you need a lot of data to notice there's a problem?
Daphne is asking if this is something we could figure out in a pilot. I think it's something we could figure out in a pilot, and it's on me for not noticing it after the pilot stage, because I bet if I go back and look at the data, it was there as well. Because something like this sort of luminance response where I know the fixed timing, it wouldn't have changed from the pilot stage to the final data set. And like I said, this data, you can already see right here, there's going to be an effect. There's a difference between the 2 lines, our 2 conditions, but it would be better if it didn't look like this leading up to the data. And so these are the sorts of things where you learn as you go, you develop new paradigms, and you tweak them and make them better in subsequent experiments. Yeah.
Could you guess from this graph where does the pupil come back to baseline?
The question is about where the pupil comes back to baseline. So this is an interesting question. So this is another where, actually, I'm going to hold this for one moment because I'm going to put up another slide that might help address this question, which was about the pupil coming back to baseline. So just hold one moment so I can explain this. A really common design that I've used in a lot of my work for a sentence listening task, we have what's called the baseline period. This is where we're getting the measure of the pupil before the stimulus actually happens. Then we have the stimulus period, and you'll notice here for my designs, I'm able to keep the visual field exactly the same. Nothing has changed. What is happening here is a sentence is playing. It's the gray mouse ate the cheese, is playing over headphones. I then, in my experiments, often have a delay period. It doesn't need to be 3 seconds. This is just something that was fairly standard in previous papers that I was building on. So I matched it. And then I have a response period. So when we think about the pupil coming back to baseline, what can be tricky in our experimental work is even though the stimulus that I am evoking a response with up here on this second screen, even though it is finished, there is another demand on the participant upcoming. And participants will also anticipate, they know what's coming in the task after the first few trials. And so they know they need to, for example, in this task, they need to remember the sentence and either type it or repeat it aloud, depending on the exact experiment. And so what this means is you might not see the pupil return to baseline during the period between your stimulus and the response. And if you go straight from the response into your next trial, then yeah, will you ever see it return to baseline? This is where I have 3,000 milliseconds here on the screen right now. I, in my recent experiments, have been pushing this out to 5 or 7 seconds. Basically, every time I collect data, I'm like, longer, more time. But this is tricky. If you want to do something like 100 trials, the difference between 3 seconds at the beginning and 7 seconds at the beginning, that actually does add up. And we'll talk about this later after the coffee break. Time within the experiment is going to fatigue your participant, and that's also going to affect the pupil. So many competing demands on trying to control for things, essentially, that we have to find the balance between them. There was another question that I asked. This is something I think to look at in pilot data. The minimum I have done is 3 seconds. The first paper I did with pupillometry, I did 3 seconds. It was what a previous paper that I was sort of building on for my master's thesis had done. And every paper since then, I have done 5 seconds minimum usually. Every task is going to be a little different. And you'll see some examples of pupil data on Friday during our sort of like mini conference. Hopefully that gives you an idea of what some other people are doing for different kinds of research too. To wrap up this slide, the critical data collection window, for example, this is just an example of my own work, and it could vary depending on what your task demands are, but it's during this baseline stimulus and delay period. I also, in all of my pupil experiments, whenever I can, I tell participants to reduce their blinking during the critical window. This is a really common thing that most researchers doing cognitive research do. It's not always something you can do with your task design. So in this case, I can tell participants, when you see a plus on the screen, reduce your blinking. This period is only going to last 10 seconds. So do the best you can. It's okay if you blink a little bit, but just try to reduce it comfortably. And then I tell them, when the O appears on the screen, blink as much as you want. We aren't collecting data anymore. Even if we are collecting data, I tell them we're not, because it's just the easiest way to explain to a participant. But in some experiment designs, you can't really distinguish between the data collection period and off the data collection period. And so you do not have to give this instruction to your participants. You will just lose more data. There will be more blinking during this critical window. And when you see data tomorrow and look at the sort of pre-processing steps, you'll see sort of the consequences of lots of blinking versus not a lot of blinking. And so for some experimental designs, there's going to be more blinking because you can't control for this. That is okay. You just might have some participants who have to be excluded from your final data set. So again, pros and cons. Any questions before I move forward? I want to show you an example of a very well-controlled study. So this is a study that was published in 2015 by Wynn and colleagues. This is a speech perception study where they were looking at the impact of what's called noise vocoding on cognitive load, and they were doing this with pupillometry. So this is the first time someone had looked at noise vocoding with pupillometry, and we'll see in a moment that their data is beautiful. The aims of their study, they wanted to see how spectral signal degradation. So I'll play an example for you in just a moment to sort of demonstrate what this is. But this is an effect where we're degrading the quality of the auditory signal. And this is going to make speech perception harder. And they also wanted to look at sentence intelligibility. We will circle back to that. They were playing noise vocoded sentences for participants. They're normal sentences like the gray mouse ate the cheese. And this noise vocoding, what it does is it simulates the resolution of speech that is typically heard through cochlear implants. So I will play this example. This example is essentially going to go through many different rounds of playing the same sentence for you with a really, at first, like a lot of noise vocoding where it's really hard to understand, and it'll lessen up over time. Go ahead, and when you're listening to this, raise your hand when you think you're able to actually understand the sentence, the words in the sentence.
What kind of bait do you use to catch salmon?
And
What kind of bait do you use to catch salmon?
Sorry, that's the next part of the example on YouTube, which is playing music through it, which is fun, but we don't need to listen to it right now. Okay, so That's what noise vocoding is. Like I said, it's meant to simulate cochlear implant audio quality, which allows us to do research about cochlear implant patients and compare them against healthy controls and sort of see if we can essentially learn from healthy controls or normal hearing controls. So what you just saw, let me move that out of the way. Nope. Oh no. Yeah. Preview. Okay, so less difficult is going to be the 32 channel. And I'm saying this also based on behavioral data. A lot of this sort of like first time doing it with pupillometry, it's following up years of behavioral data in lab. And so we have good reason to have strong hypotheses that the least difficult of these should be the 32 channel. There's the least amount of noise vocoding here. There's much more on the 4 channel. And so if we look at the data, what we see is this very nice hierarchical relationship. The pupil response, the amplitude, this how high this peak of the response goes is higher up as the speech is more degraded. They had a second aim in this paper that I also want to talk about just because it's very interesting for my own work, and maybe there are also applications outside of speech perception. What they did with this analysis, so this is the same data set represented 2 ways. So the plot on the left here is what I just showed you. It's the same plot. And now I have on this right side, they took only data where participants made a correct response. So what do I mean by that? They're listening to sentences like the gray mouse ate the cheese. On the left is all of the data. So some of these sentences, when the participant had to repeat aloud, they said the gray something ate the something. They didn't understand it because it was really hard, as you saw with the noise vocoding. On the right here, we are only plotting data when they were able to correctly repeat back the sentence. And for all of these levels, so there is some amount of data, probably much less for the 4 channels, which is very hard. And what we see is this hierarchical relationship remains. There's a little bit of differences. You can see in like how the slope after the peak looks. Don't worry too much about that for now. The big picture thing that I want to convey here is that we can conclude that the effect of noise vocoding is present in both analyses, meaning that the pupil response can track cognitive load even when performance is at ceiling with a behavioral measure. So that behavioral measure was sort of like the very typical for noise vocoding research, just how many words were you able to understand? And you would hit ceiling where people are getting all of the words correct. But just because a participant is getting all of the words correct, they're able to repeat back the gray mouse ate the cheese, that doesn't mean that they didn't put in more work along the way to get to that correct response. This is something that I think is really powerful about pupillometry. You can look at sort of a typical behavioral measure and you can ask the question, okay, but when people hit ceiling, when they start to perform really well, can we still see differences in performance at that level? And this is, I have used this a lot in my own research because I look at perception of accented speech. And a lot of the times accents are highly intelligible, meaning we can understand them, but our speech processing system might be putting in a little more work along the way in order to understand them. Okay, we're almost done with this session. I know a coffee break is very due. Those are the top 3 nuisances. These are the things in particular that good cognitive pupillometry research is going to control for. You won't have these happening at the same time as your evoked response, and that'll give you the cleanest data. It'll help you draw the most firm conclusions from this type of data. I have a group exercise, but let's wait until after the coffee break because, yeah. Oh, and Manu?
You have to look at
Mhm.
If you go back to the so do you have the same for errors?
No, I'm.
Errors triggers the ACC. And
Mm.
if the ACC triggering the pupil response, then I'm expecting a bigger response for errors.
Yeah. Manu is mentioning that the ACC is triggered by error response. I love this thought process and I've never thought about it before, but yes, in my own work, you do see when participants can't understand a sentence. So this example on the left, you see larger pupil response. It's like called an intelligibility measure. So if there are more error signals, there's larger pupil response. But I have never heard anyone describe it that way. And this is why we all need to come together and talk about our research together.
Then I have a curiosity.
Yeah.
Once is number 2. Do you know any study which about pupil response for imaging? So I'm image, so think about a square that I know very well, and then to change my perspective inside the square, but only in my mind.
In your mind, yes. So Manu was asking whether thinking about something in your brain can elicit a pupil response, essentially a visual thing. Yes, there is actually data for the pupil light reflex, for example. If you tell someone to picture a bright room, the pupil will constrict. So this also, I know, there are entire groups of people who do mind wandering research with pupillometry, which I don't know how they do that. It sounds chaotic, but yeah. Daphne?
I don't want to keep this from coffee. That made me think of something. So do emotions also affect it? Like if I'm really annoyed by that or vocoding thing, like is my pupil going to change in response to that? Or if I'm surprised by it?
So Daphne's asking about emotion and surprise. I don't know much about emotion, but I do think there is pupillometry research in emotion. I think it exists. That's like a field. I just don't know anything about it personally, so apologies. Surprise, yes. In fact, there's attention researchers would probably argue that surprise is a large part of the pupil response. But I think as I was trying to sort of demonstrate with that taxonomy, it's very complicated, it's multifaceted, and all of these things are happening at once and competing to affect the pupil one way or another. So yeah, surprise is something that plagues me a little bit in my work because I think about like, do I want to tell the participant exactly what to expect or not? And I typically err on the side of preparing my participants for the experiment because I don't want surprise-based arousal mixed in there, but there haven't been very firm methodological studies. So when we come back from the coffee break, we're going to talk about what we don't know, which can be very frustrating, but also informative. So coffee.
Is recorded. And I also wanted to tell you that I'm not in the mailing list.
Oh,
So,
sorry.
no, don't worry. I asked Julie to send it to me.
How do I, okay, so